Welcome everyone to Iris Biotech's fourth online workshop. True to our slogan, Empowering Peptide Innovation, we are keen on knowledge exchange and getting in contact with scientists all over the world. Therefore, we started this workshop series last summer. Today, we are focusing on peptide EasyClean technology and its use for the synthesis, modification, and purification of difficult peptides. My name is Raimund Meyer. I am working as a project manager at Iris Biotech, and today I will guide you through this online workshop. If you wish to announce your participation on social media, please feel free to use the hashtag Iris Workshop Series, which is also displayed right now in the Microsoft Teams chat. To have a pleasant experience for all participants, I would like to begin with some technical guidelines. To ensure the best possible audio and video quality, all participants are automatically muted and your video camera is switched off. Another important information is that this live event is recorded. Thus, if you enter late or miss anything, please check the bottom left side of your Teams screen if you are watching the recording or if you are live, which is shown by the red broadcast symbol. You have the possibility to pause the video, rewind, or jump back to the live session. In addition, the recording of the workshop will be uploaded on our homepage afterwards. Questions can be submitted by using the Q&A chat of Microsoft Teams. You can enter your name or submit your questions anonymously. We will frequently check the questions, publish them, and submit them to the respective speaker. To try out this Q&A chat, we will now post a question which you can answer. So now you should see the question, what does the abbreviation PEC stand for? Within the chat also. Please feel free to answer it to try out this tool. If you have any technical queries, you can also submit them in this Q&A chat. Our technical assistant will then connect with you and try to solve the problem. Initially, the questions are just visible for the organizers and will then be published to the audience. So don't worry if your question takes a short moment to appear on the screen. We now see the answers are coming in, and yes, of course, the correct answer is PEC stands for Peptide EasyClean. So as this seems to work, I would like to quickly give you an overview of today's agenda. We will start with a short company introduction by our project manager Karin Rustler. Afterwards, we will pass on to our experts from Bilintic. First, Nadia Berger will focus on a joint case study on the peptide liraglutide. Afterwards, Manoj Kumar Mutiala will present general considerations for PEC on large scale. As mentioned, questions will be collected throughout all talks in the Q&A chat and are forwarded to the speakers. Of course, you can also get in contact via email or phone call after the workshop. One last note from my side, if you accidentally close your Microsoft Teams window, don't panic. As the workshop is also available as recording, you won't miss anything. Just re-log in and view the recording. You can then also play this recording at increased speed until you catch up with the live event again. After this quick technical intro, let's continue with the first agenda point, a short introduction to our company by our project manager, Karin Rustler. Karin studied biochemistry at the University of Regensburg and obtained her doctoral degree in organic chemistry for the synthesis and characterization of photoswitchable compounds for biological applications. During her PhD, she stayed abroad at Kyoto University, Japan for several months. Beginning of 2020, she joined Iris Biotech as a project manager responsible for customers in Spain, Portugal, Japan, and Korea, and she is in charge of our marketing activities. Karin, thank you for your precious time. Please go ahead with your presentation.
Thank you, Raimund, for the kind introduction. I hope you can see my screen now. And with a few words, I would like to briefly introduce the company Iris Biotech to you. Our roots date back more than 200 years to the chemical factory Marktredwitz. In 2001, Iris Biotech has then been founded to strengthen the market position in the field of peptide chemistry. Over the years, we built up a worldwide network with cooperation and distribution partners. Due to the growing demand for custom products, we founded the Iris Biotech Laboratories in 2013. On the occasion of our 20th anniversary last year, we refreshed our corporate design and switched to a new company logo. So here shown on the right side, you can see the new logo in comparison to the old one shown above. And what you can see in the background of the slide is our company building. Following our slogan, Empowering Peptide Innovation, our mission is to support our customers as qualified supplier and competent consultant. Thereby, we are covering requests from research scale to commercial productions. Our portfolio includes products for peptide synthesis, modification and purification, life science products, as well as technologies for linker construction and drug delivery. Besides, we are frequently publishing our newsletters and self-made brochures, which are available as free download on our website. To be able to provide innovative products and latest technologies to the whole peptide community, we are working together with universities and companies worldwide and are thus serving as a distribution and networking platform, not only for products, but also, and most importantly, for knowledge exchange. With this, I would like to introduce to you our cooperation partners at Bilintic. We are working together for around 2 years now. And for me, this is certainly some sort of special relationship, because before I started at Iris, during one of my job interviews there, I had to present self-chosen innovative products, which I thought would fit to the Iris portfolio. And the product or technology I chose was the Bilintic's Peptide EasyClean, which was then also the first project I worked on when I started with Iris. So with this, it's a real pleasure for me to pass on to Bilintic, and I'm looking forward to your talks.
Yeah, thank you, Karin, for giving this introduction and also for these personal remarks. So let's now switch to our external speakers and their presentations. It's my pleasure to introduce to you Nadia Berger of Bilintic. Nadia studied chemistry at Cologne University and received her doctoral degree at the Ruhr University Bochum, including some research time at the University of Queensland in Brisbane, Australia. After postdoc time in Pennsylvania, she joined the team of Bilintic in 2019 as an R&D peptide chemist. She has more than 10 years of experience in peptide science. At Bilintic, she's mainly focused on the development of the peptide EasyClean technology for specific applications, particularly the manufacturing of modified peptides. In this regard, she is now scientifically leading the development of Bilintic's platform PEC. Today, Nadia will present our joint results on the synthesis and purification of liraglutide. Nadia, it's a pleasure to have you with us. The stage or video platform is yours. We are looking forward to hearing about your research. And for the audience, please post your questions by using the Q&A chat again.
Thank you, Raimund, for your introduction, and welcome everyone to the first part of this workshop, where I am first going to introduce you to our peptide EasyClean technology, and will then continue to talk about the use of it in the production of liraglutide. Bilintic's proprietary peptide EasyClean technology is a novel multi-feature tool to address unmet needs for peptide drug development and production. It's the first-in-class parallel purification tool for fast and automated manufacturing of peptide libraries. In addition, it allows to integrate peptide modification to rapidly create new peptide leads. And it enables the production of complex peptides at large scale or in a fast, green and efficient way. For the peptide purification, you need our PEC linker, which you see on the slide. It's a small molecule and actually the first universally applicable catch and release linker. It's site reaction free and available in kilogram scale for industrial processes. The linker molecule contains 3 main parts which are necessary for the PEC purification. The first site here, which is important, is the Boc-protected oxyamine, which serves as catch tag for the immobilization via an oxime ligation. Then in the middle, you see an aryl azide core, which is a reductively cleavable unit. And on the right side is an activated carbonate with a leaving group for the coupling of the linker with the N-terminus of the peptide. Now I will introduce you to the way it works. First, here I will give a brief overview, and then on the next slide I will show some more details of the procedure. So the technology is used for the purification of chemically synthesized peptides by SPPS. And then in step 1, or actually the last step of the SPPS, the PEC linker, which is shown here as green circle, is coupled onto the full-length peptide before the peptide is released from the solid support in step 2 by TFA cleavage and precipitation, and also the side chains are deprotected in this step. Then in step 3, the actual purification starts. Here the peptide is dissolved, and of note, you are really flexible in the choice of solvent, which is a significant difference compared with HPLC, where you are really restricted to the solvents that you can use for the purification. So now that the peptide crude is dissolved, it's added to a fritted card holds some activated filter material. This filter material is or are aldehyde modified beads with a shelf life of more than 2 years and a high capacity with 60 to 100 micromole per milliliter settled beads. This means that there is basically you can process a lot of materials or a lot of peptides with the use of a low amount of beads. And with this, you can also process a lot of material in small containers. So after you added your dissolved peptide to this cartridge with the filter material in step 4, the peptide is now caught or immobilized onto the solid support via coupling of this linker with the material. Then step 5 is a washing step where all dirt from SPPS is removed and only the full-length peptide stays in the cartridge. Step 6 is a optional step where you can modify the peptide via side chain modification, and my colleague Manoj will talk about this later in more detail. And then in step 7, the peptide is acidically released from the solid support, and you will obtain it in its purified and potentially modified version. So now let's have a closer look at the whole process. So you start with the coupling of the amino acids one after another onto the SPPS resin to give the full-length peptide, which is side chain protected. And between each amino acid coupling follows a capping step with acetic anhydride, so that all truncations are kept, whereas the amine of your desired peptide remains free to be able to react with the carbonate group of the linker in the first step, the PEC linker coupling. Then follows the TFA cleavage, which will release the peptide from the solid support and deprotect the side chains, but also this Boc-protected oxyamine here, which then allows, after dissolution in step 3, the immobilization in step 4 by reaction between the oxyamine and the aldehyde of the filter material in an oxime ligation. Then follows the washing step, where you can wash away all truncations or other SPPS dirt so that only the linker coupled peptide stays in the cartridge. And then I will skip the part with the modification. So in step 6 is here now the release of the peptide from the activated material. And this is initiated by reduction of this A site here to an amine. Then the reducing agent is washed away, and upon acidic treatment, a 1,6 elimination as indicated here is initiated and the peptide is released from the solid support. So in this workshop, we will give some highlights of PEC. First of all, I will talk about the processing of difficult peptides, which is made easy by high solvent flexibility. And then I will show you how easily the chemistry is scaled up. And then in the second part, Manoj will introduce you to fast access to modified and purified peptides in a combined approach. And then he will present or give some consideration towards next generation manufacturing of complex peptide APIs. So now I will talk about the easy access to purified complex peptides on larger scales with the Peptide Easy Clean technology. Using the example case study of liraglutide. If you are interested in more details, you can find our article on this case study in Chemistry Today, which we published together with Iris in the end of last year. Liraglutide is a long-acting analog of a GLP-1 with a fatty acid side chain on the lysine residue at position 26, as you see on the right side of the slide. It is marketed under the brand names Victoza and Saxenda by Novo Nordisk for the treatment of type 2 diabetes. And liraglutide is a blockbuster drug with sales of more than $4,000 million in 2019 alone. However, its patent expires in 2023 in the US and in Europe, which is why it becomes very attractive for the generics market. And the most efficient manufacturing processes are desired to compete here. However, there are some challenges in the liraglutide manufacturing, which are basically mainly due to the lipid on the side chain of lysine. So the typical preparation of liraglutide can be either by orthogonal side chain protection and on solid support modification, or by modification in solution after the SPPS or after the recombinant synthesis as done by Novo Nordisk. Because of the lipid, the solubility of liraglutide is very poor, especially below pH 7. And it is very prone to aggregation, which makes the handling difficult and especially complicates the purification process. So we at Bilintic developed a new approach for the production of liraglutide where we combine SPPS with PEC. For the SPPS, we use pseudoprolines. This was already done before, and it showed that the crude purity of liraglutide is significantly increased when you use these 3 pseudoprolines. And then we used a lipidated F-morglycine building block, and all of these building blocks are available at Iris Biotech. The synthesis of the peptide was done on a pure PEP chorus synthesizer. First, we synthesize 0.5 millimole in batch 1, and then we synthesize 4 times 500 micromole of the peptide. After that followed the PEC purification at different scales. So we did it 1 times with 220 micromole, then with 200 micromole, and in the end with 2 millimole of the crude. And we were especially interested in solving the solubility issues with liraglutide. So typically when we apply our standard PEC protocol for the PEC purification, we use the MSO as solvent to dissolve the peptide before it is immobilized. This helps already to get a lot of material usually dissolved, even a bit more rough cases, and is a benefit compared with HPLC, where you are usually restricted to a minimum of peptide that you can purify at a time. However, for very difficult cases such as liraglutide, you may use an alternative solvent. And here we used 2-to-1 mixture of HFLP and saturated aqueous guanidinium chloride, because the solvent and this chaotropic salt are able to further increase the solubility and prevent and break aggregates. So with this, we applied our standard protocol first for a small scale where we purified 20 micromole of the compound. And we did this in these kind of fritted cartridges that you see here on the left side, which is basically an SPPS reactor and which is available in our kit products as well. And after PEC purification, you get your nice purified product. Then we applied the same protocol to purify 200 micromole just by the use of a bit of a larger reactor. And then we did the purification of 2 millimole peptide using standard lab glassware. As you see here, we just use a round bottom flask with a mechanical stirrer for the immobilization, for example, and whereas the filtration tasks were done using just a glass frit. And here I want to point out that you can basically use any kind of reactors, also SPPS reactors, when you go to even larger scales. And my colleague will also introduce a new way that you can use to purify a large amount of material, but you don't really need any special equipment if you don't have it available. So let's have a look at the results. First of all, when you see at the crude from batch 1, you see that we were able to achieve a purity of 61%. And after a single PEC purification of the 20 micromole, we could increase the purity by more than 20% to 85%. When you now have a look at the UV chromatograms of the after purification of 200 micromole and then of 2 millimole, you see that there is basically no decline in purity, which means that there is no difference in PEC purity between the individual runs at various scales. The same was basically obtained in terms of yield. So when you look at the final amounts that we were able to get after purification of the individual scale, it was pretty much the same. And the recovery was also retained throughout all 3 scales. So finally, I want to give some large-scale considerations here. So we did some calculations in order to see what's in it for liraglutide when we want to achieve an API grade purity in the end, where we would combine PEC with HPLC. So in this calculation, we start with 1 kilogram crude material, where we assume that it would contain 60% peptide. This would mean that no matter how you purify the material, you could theoretically end up with 600 grams of pure product. And now we calculated once the process for a combination of PEC with an HPLC run, where you could simultaneously also purify the material, and compared this to a process with 3 HPLC runs as typically required for the purification of liraglutide. So for the recovery, as I showed you before, we were able in our case study to recover 50% of liraglutide, but typically we see for other peptides a recovery of 80%. And we believe with when we fine-tune our purification process, we could recover 80% of liraglutide. In the final HPLC polishing step, we assume that we could recover 90%. And this would mean that we could end up with 432 gram of purified liraglutide. On the other side, when we do the HPLC purification, we assume that for a heavily overloaded run, we could recover 70% of liraglutide or product in the first run, in the second run 80%, and then also 90% in the last step. This would mean that you would end up with 300 gram of final material. And when you want to obtain 1 kilogram, it means that you could potentially save 50% of the cost when you combine PEC with HPLC. So you could really drastically reduce the overall API manufacturing costs. So in conclusion, I hope I could convince you that PEC enables the manufacturing of complex and hydrophobic peptides with a flexible choice of solvents and an orthogonal approach, with the processing of large amounts of material in only small containers. The scale-up of peptide purification with PEC proved seamless and simple without any decline in efficiency in our case study for liraglutide. And the unique catch and release method allows a flexible use of cartridges and reactors of basically any size. The process holds promise to significantly reduce the overall costs of API production. And with this, I'd like to thank you for your attention and yeah, hope you have some questions.
Yeah, thank you, Nadia, for sharing these results with us. Indeed, we did collect some questions. The first comes from David Fercher. Can this technology also be used when the final peptide needs acetyl capping?
It depends on the peptide. So typically it requires an amine that is free, and you could potentially do it via side chain. So that you, when you, for example, have a lysine on your side chain and use orthogonal protection, then potentially you could there equip the peptide with the linker and then have your final N-terminus acetylated. But usually you, or at least you definitely need a free amine.
Yeah. So if you have the option to attach the linker via sidechain, yes, but generally rather not. Okay.
Yes, exactly.
Then the next question, what should I keep in mind? What kind of precautions are necessary for a PEC run?
So yes, in order to really get the highest recovery possible, you certainly should avoid to use any kinds of solvents that could contain aldehydes or ketones, because they could react after the deprotection with the oxyamine, and this makes the linker coupled peptide unavailable for reaction with the aldehyde on the activated material. And what's probably also crucial is let's say, good shaking or rotation of your beads during the immobilization process.
Okay. The third question, is there any restriction regarding the peptide length that can be purified by PEC?
No, it's not. And actually the PEC purification becomes more and more attractive, I would say, for longer peptides, because it becomes more and more difficult to remove truncations which are similar to your desired peptide to purify it by HPLC. And whereas with PEC you can still remove these truncations. And yeah, for those who are interested in more details on that, you could have a look at our case study difficult peptides, which you will find on our website. But it's really specifically good for longer peptides, I would say.
Okay, then a question to the recycling. Is it possible to regenerate the filter material?
No, at the moment it's not.
Okay, then are there special coupling conditions for the linker?
Basically, you can handle it as any other amino acids. So typically we use oximer for activation, and yeah, no, there's nothing special needed.
Okay, and there is one question that already appeared in the same way by Ganesh. Is the PEC resin reusable? If yes, for how many cycles? You already answered this. It's not possible. So because when it's reactive.
I mean, just of note. I mean, potentially you could probably somehow get the aldehyde groups back, but I think it would be difficult, and so yeah, right now it's not possible.
And then one more question. Thanks for the presentation. Is there a risk of imine formation of the aldehyde resin with lysine side chains?
No, not that I would.
I think there is. I can answer this question, I guess. There is a possibility of immune formation, but we block the aldehydes by cysteine after the immobilization. So there will be no aldehyde present.
Okay.
So yeah, the imine will be reversible reaction. So the cysteine will knock out the imine and form a cyclized product.
So you have an excess of the cysteines, which will definitely say open up all imines.
Yes.
Yes.
Yeah.
Okay. So currently we do not see any further questions within the chat. Of course, if you have any more, do not hesitate to contact us by mail or phone, and we will forward those questions to our experts. Nadia, thanks again. It was a pleasure to have you as one of our guest speakers. And now I would like to introduce to you Manoj Kumar Mutyala. Manoj studied chemistry at the JNTU in Hyderabad, received his PhD at the Birla Institute of Technology and Science in Pilani, Rajasthan, followed by a postdoc position at FU in Berlin. He joined Bilintic as a research chemist in December 2018 and has a strong expertise in synthetic organic polymer peptide and carbohydrate chemistry. In addition, he has 3 years of experience with the PEC technology now. Currently, he is exploring feasibilities of PEC as an advanced modification tool for late-stage peptide modifications. Manoj, we are looking forward to hearing more about your research on PEC for large scale. We're happy to have you with us. The platform is yours. And for the audience, please post your questions by using the Q&A chat again.
Thank you very much for the kind introduction. Yeah, title of my talk is Disulfide Formation and Step Towards the Next Generation of Peptide Production with PEC. I have divided this topic into 2 subtopics for the easy understanding. In the first subtopic, I will be discussing about how can PEC is useful to access modified and purified peptides in a combined approach, where we use this approach to manufacturing of disulfide peptides. And then the second approach, I will be discussing about Bilintic step towards the next generation production of peptide APIs. How we want to achieve this. We want to use by these 3 methods using SPPS, green SPPS method, direct cocktail immobilization, and scale up in rotating bed reactors, RBRs. So coming to my first topic, fast access to the modified and purified peptides in a combined approach, I want to give you an overview how we can modify peptides during our PEC technology. During SPPS synthesis, amino acids are coupled to the resin. After each coupling, capping was performed to prevent the growth of the truncated sequence. At the end, our linker molecule is coupled to the N-terminus of the peptide, which upon TFA cleavage will release the crude peptide along with the truncated sequences. This is precipitated by ether precipitation, then lyophilized, and again resolubilized based upon the solubility of the crude mixture. Then this was treated to the modified aldehyde modified beads. Only linker coupled peptide is covalently bound to the modified beads, whereas the truncated sequences can be easily washed away. This gives us a clear window to perform various modification reactions. Then final release will give us purified and modified peptide. So where can it help? It can help in doing pegylation reactions. It can help in cyclization reactions such as disulfide formation and stapling. And it also helps in biotinylation and lipidation reactions. So what are the benefits of this technology? This is highly chemoselective methods, and we can modification and purification achieved in a single step. Here I would like to mainly focus on disulfide formation, taking an example of solanitide. Solanitide is a 17 amino acid peptide with one disulfide bridge. Severely affected COVID-19 patients suffer from acute respiratory distress syndrome. The solanitide is helpful to remove the water from the lungs and also helps in the oxygenation. This helps reduces the need for mechanical ventilation. So how does the PEC technology works in the late stage modifications? Here I would like to give you a deep dive into how our technology can be useful to synthesize disulfide peptides. Here we can use any different protecting groups using our PEC technology. For example, we can use trityl cysteine protecting groups, or we can use tertiary butyl protecting group to synthesize disulfide bonds. Here in my case, in the solanitide case, I used trityl protecting group to prevent beta elimination followed by piperidine attachment at the C terminal of cysteine. One can use other protecting groups like tertiary butyl if there is no cysteine present at the C terminus of the peptide. Upon linker coupling and TFA cleavage, we could achieve the linker coupled peptide, which is cleft from the SPPS resin. During this one, if the trityl protecting groups are used in the cysteine, it will be cleft off. Then the linker coupled peptide is immobilized on modified PMA beads and then reduced using DTT in the presence of basic conditions, where aryl azide core is reduced to aryl amine core. Still, this linker coupled peptide is still intact in the modified PMA beads. This gives us a clear window to perform disul oxygenation reaction such as disulfide formation. On the other hand, if you use tertiary butyl protecting group here, this will be also cleft off during this reduction step. As I told you, this is quite stable molecule. We can perform the oxidation reaction and we can achieve the disulfide bond here. And then finally, using 95% TFA, we could achieve the desired solanitide in a pure form. In the next slide, I would like to compare how does the PEC compared to the solution phase oxidation. So here you can see right hand spectrum. The solanitide can be purified, PEC purification without oxidation also. The purity increased from 71 to 83%. Whereas if you perform the oxidation reaction during the solution phase, the purity we could achieve only 46% purity of oxidized solanitide. With dimer formation and sulfenic acid, these are the main impurities we observed in a solution phase. Whereas if you perform the oxidation also in the PEC, the modification, as I showed you in the previous slide here, oxidation during the PEC reaction, we could achieve the disulfide bond purity of 75% compared to the solution phase, where we can achieve only 46%. Here we can completely mitigate the dimer formation, as you can see here, and we can completely reduce the sulfenic acid formation, which is over oxidation. The reason is PEC. This is in the PEC, that pseudo dilution of PEC eradicates the dimer formation and decreases the sulfenic acid, over oxidation and sulfenic acid. So what are the benefits of this process? This is we can achieve the fast disulfide in a short period of time. Within 6 hours, we can get our purification, oxidation and purification is finished. And this is whole technique is purification and modification is a combined approach. So I hope I convinced you that with our modification technology, one can synthesize a complex peptide like solanitide, where we can modify and purify in a single shot. Next, I will come to the second topic of my presentation, where the Belintic steps towards the next generation of peptide production. Our Belintic is committed to sustainability. It promoted us to work in the field of green chemistry. So our goals are, Belintic goals are using green alternative solvents for SPPS and PEC and decrease the solvent consumption. And we also want to provide a seamless, robust, efficient scale-up reaction, which can synthesize peptides in multi-kilogram scale in a closed containers. So to achieve this, we have revisited each process from starting SPPS synthesis from the till the purification, and we came with three possible approaches where we can improve the process and we can give you the next generation of peptide production whole package. So the first production is we want to focus on green SPPS synthesis, and second one is direct TFA cocktail immobilization, and third one is I will tell you the possibility how we can scale up the PEC, purify the peptide in multi-kilogram scale. So coming to the first green SPPS synthesis, we have taken here bivalirudin as an example. So the whole idea is using a green binary solvent mixture in the SPPS synthesis alternative to the DMF. DMF is proven to be toxic, so we want to use green binary solvent mixture. So we are inspired from the Martin et al. Group published in the Green Chemistry in 2021. So what the key findings from this publication are, the most important is the solvent viscosity and polarity plays a major role in the purity of the SPPS outcome. And even higher polarity increases the FMOC deproduction rate, whereas the lower polarity increases the coupling rates. And lower polarity decreases the even aspartamide formation. So we want to take this consideration key findings and we want to apply our experience, SPPS experience, and we want to modify and we want to make it a little better. So, and we want to see that after synthesis, green synthesis, whether our PEC process is compatible with the green SPPS or not, whether we can purify the impurities, we can purify the bivalirudin by removing the impurities or not. So as I told you, we have taken the consideration from the publication and we have, based on our SPPS knowledge, we have even modified the procedure. What we have done is we have took the washing solvent DMSO ethyl acetate 2:8, whereas in the publication by Bahem they used isopropanol. For the FMOC removal, we used same solvent as the Bahem, which is used as the DMSO 6:4, DMSO ethyl acetate. We want to use this same solvent mixture, green solvent mixture, because and the high polarity solvent mixture. The reason is the piperidine removal of FMOC removal produces the anion, which will be stabilized by high polar solvents. On the other hand, we want to use amino acid stock, higher concentration of amino acid stock solution with 0.5 molar compared to the reported methods. The reason is we are forced to use here high polar solvent compared to the previous procedure reported 2:8. The reason is only DMSO 4:6 can solubilize high polar, high concentrated amino acids. To decrease the final concentration in the coupling reactions, what we have done is we have taken the DIC in ethyl acetate and oxymot solution in 4:6. What happens in the coupling reaction, obviously everyone knows that the DIC and oxyma will be added to the amino acid. So the final coupling mixture would be, it reduces the polarity from 4:6 to 3:7. And capping was performed in 1:9 DMSO ethyl acetate. And finally, we performed isopropanol used for alternative for DCM for washing. The whole synthesis was carried out in PurePep chorus synthesizer, as you see shown in here, and 0.5 millimole scale. So we could see in the right-hand spectrum, right-hand side chromatogram, we could not only reproduce the result, we even got the better purity. We could increase the purity from 74% to 74% here, compared here 88%. There are some impurities with truncated sequences, deletion, and aspartamide formation. Now we want to check whether our PEC technology is compatible with the green SPPS or not. So what we have done is we tried to purify the bivalirudin crude material using our PEC normal PEC procedure. So what we do here is after linker coupling to the SPPS bivalirudin, we cleft off using TFA cocktail and then we precipitate it and then we again resolubilize it in HFIP. Then the PEC process was continued. Here you can see the purity was increased from 88 to 93%, and we can successfully remove all truncated sequences. Yeah, the purity is increased from 88 to 93%. You can see we have even saved the 75% of solvent consumption compared to the HPLC, which can be, yeah. And then there is still, based on this example, we can clearly say that our PEC technology is compatible with the green SPPS reaction. And there is still still possibility in the improvements where we can fine tune the solvent polarity by changing the solvent polarity, and we can even decrease the impurities like aspartamide formation and deletion peaks, and we can even achieve the purity more than 95-96%, which API grade. So with this, I would like to go to the next step towards our Belintic step towards next generation of production peptide APIs. So this is direct TFA cocktail immobilization. So generally, as my colleague Nadia explained you, we generally use DMSO or HFIP for solubilizing the linker coupled peptide to immobilize on the filter material after SPPS synthesis. So DMSO works quite good. HFIP also works good. The problem is DMSO may oxidize the methionine and cysteine. And on the other hand, HFIP is costly and volatile. So we thought we are using anyway TFA in the final process, in the PEC process, to release the peptide from the filter material. We thought, why can't we use TFA for immobilization, immobilization solvent? So we thought because TFA solubilize every peptide like hydrophobic, whether it is hydrophobic or whether it is hydrophilic. So then we thought of investigating TFA as a solvent for PEC immobilization. And then the main question occurs here, whether our oxime ligation linker coupling will happen on the solid support or not. And the second, if it works, can we directly immobilize TFA cocktail immobilization just bypassing the ether precipitation as shown it here? So step by step. So first we tried TFA immobilization. What we had done is after SPPS synthesis, as shown it here, we cleft off the peptide, linker coupled peptide, and we solubilized, we precipitated it and we solubilized in TFA, and then try to immobilize on agarose beads, modified agarose beads. Then we later realized that the modified agarose beads are not quite stable to the TFA treatment, higher concentration of TFA treatment due to less mechanical strength at a high pH. Then we were searching for alternatives. We found that modified PMA beads are quite stable and quite robust, which can withstand the TFA up to 95%. So then we tried to immobilize our linker coupled peptide on modified PMA beads by solubilizing linker coupled peptide in a TFA. It worked quite nicely. The immobilization worked quite nicely. Then we thought, why can't we use direct cocktail, just avoiding all the precipitation step, directly immobilizing the cocktail mixture to the filter materials. We are happy to share you that the direct cocktail immobilization using TFA TIS water as a cocktail successfully immobilized the PMA beads, bypassing the ether precipitation step. We could successfully purify the peptide in a good scale. But we later realized that, however, due to the TFA immobilization and direct cocktail worked quite nicely, we see that immobilization is little slower, and we see that observed that recovery is also slightly lesser compared to our HFIP immobilization. Then we thought, what's happening? So then we came across that our linker Bock protected oxyamine will be cleft off in a TFA cleavage and forms ammonium ion, oxyammonium ion in the TFA conditions, which will be protonated. It is difficult to react with aldehyde groups when it is in the protonated form. So then we thought, so it's in the equilibrium state. So that's the reason the only deprotected or unprotected amine will react with aldehyde molecules, whereas protected reactions will take place, the immobilization taking place a longer time. So then we thought, can we fine tune the TFA cocktail by adding water, or by diluting with acids, or diluting with bases, or diluting with organic solvents? Then we tried different conditions. We initially tried to dilute with water. Then we later realized that if you dilute the cocktail with water, even the peptide will be precipitated out, crushed out. So using the water is out. Yeah, it's not a good idea. Then we thought of diluting the cocktail with organic solvents such as DMF and acetonitrile. And we are happy to share you that the recovery, the immobilization is faster, even if you use dilute with the cocktail with DMF and acetonitrile. On the other hand, if you neutralize the cocktail with the bases such as pyridine, has even improved further immobilization further, and the recovery was higher. You can see in the right-hand side spectrum chromatogram, I compared the cocktail when it is diluted with the DMF and when it was further neutralized with the pyridine and then diluted with DMF. You can see the internal standard here, and you can see the recovery is higher in the cocktail which is neutralized with a little amount with pyridine and then diluted with DMF. So we could achieve the good recovery and good purity compared to the our normal PEC protocol, where we use HPA, where we use different organic solvents, DMSO. Here we can directly immobilize from the cocktail by diluting with organic solvents and neutralizing with bases such as pyridine. So the optimal immobilization conditions are TFA cocktail. We can use pyridine up to 7.5 to 15 volume %. And then we can dilute this whole mixture with acetonitrile and DMF. So taking the green chemistry into consideration, we prefer to use acetonitrile rather than DMF. So then the question arises, what happens if there is cysteine and methionine containing peptides, methionine containing amino acids are there in the sequence? Can we use thiol scavengers? Because till now we used DMF, we used TFA TIS water. We didn't use any thiols in the scavenger. Currently experiments are in progress. We'll show that initial results looks quite promising, but we'll come up with soon with how we can even use thiols in the scavenger and can purify our peptides. So then coming to the scalability. So we want to use this whole PEC process, make it seamless, robust, and efficient process. We want to use synthesize, purify peptides from gram to kilogram scale, and we want to use them. The purification should be easy by washing away the impurities and solvents and reagents. So our collaborator SpinChem has a new technology which is rotatory bead reactor. What happens in the rotating bead reactor, as shown is here in the figure, is the mesh size in the rotating bead reactor is 100 microns. So what happens is the PMA beads will stay in the rotatory bead reactor, and then we can continue our PEC process using this SpinChem apparatus. The solvents the impurities can be easily washed away by here, and then we can even feed the reagents and organic solvents from the top. So we thought we use this SpinChem technology and we check the compatibility with our PEC process, because it's look like as I shown it here in the figure, we can upscale the purity from gram to kilogram scale. So we try to test the whether our PEC process is compatible with the SpinChem or not. As it's shown here, we have taken an internal standard peptide from our library, and we have performed this PEC process, and we have compared the SpinChem versus cartridge. Cartridge generally we use for our PEC purification for smaller scale reactions. You can see here the purity, we are happy to share that the immobilization is really faster in rotatory bead reactors due to efficient mass transfer. And the purity of the PEC purity was also slightly higher in the SpinChem compared to the cartridge. And due to the efficient mass transfer of SpinChem technology, we could even achieve the more recovery in the SpinChem compared to the normal cartridges. So what it means, this gives us all-in-one reactor, will dramatically save your overall time, and we can easily scale up the peptide purification from gram scale to kilogram scale. So as I told you, as I started my second presentation by saying that Bell-Intic step towards next generation of production of peptide APIs. So we want to give you, ultimately, we combine the green SPPS synthesis, direct TFA cocktail immobilization, and processing with RBR for the purification of bivalirudin to show you that we can provide you Belintic can provide you a next generation of purification technology from gram to kilogram scale. If you can see the right-hand spectrum, right-hand side chromatogram, the purity was increased from 88 to 92% by using direct TFA, green SPPS synthesis, direct TFA cocktail immobilization, and RBR technology. So what is the impact actually? So we completely removed the DMF, DCM whole in the whole synthesis and purification process, and we avoided the precipitation and dissolution step that drastically shortens time to product. And we use TFA cocktail immobilization, ensures that the solubilization of complex peptides, whether it is hydrophilic or hydrophobic peptides, TFA cocktail will solubilize all the peptides. And finally, we want to emphasize that the purification compared to the HPLC, we saved up to 75% solvent consumption. And this is robust handling in closed container for scale up in multi-kilogram scale. And we want to, in future, we want to even apply this technology for late stage modification and see the how the spin chem works, how the whole process works. So with this, I hope that I convinced that the Belintic can give you a solutions for large scale production of APIs. Thank you very much for your attention.
Yeah, thank you very much, Manoj, for sharing your results with us. And during your talk, we collected further questions. Indeed, there were even quite some questions that still referred to the first Q&A session. So I would ask you and Nadia to switch on the microphones, and questions will go to both of you. Onkar asks, please elaborate capture resin details. Is it required to pack them in the column?
No, there is basically no column required. So if I understood the question correctly, the question is where the activated filter material is held. And so, yeah, you can have them in everything you want, as mentioned, I think not shown in detail. You can even have a round bottom flask and a mechanical stirrer, or you could just add it to some cartridge that holds any type of container where you can shake or rotate, for example, when you have it in a SPPS reactor, or as shown by Manoj in this spin chem reactor. So there is no specific column or anything that you need.
Mm-hmm. So some questions coming up to the remaining impurities. That also to your talk, Nadia, why only 80% of desired peptide after release? What is the 20% byproduct? And a similar question, how can you reach higher purities then?
So it was difficult to me to figure out what the impurities in this case were exactly, especially since there was one more prominent impurity, but where I had the same or no mass that I could assign to anything specifically. But in general, it can be either that your truncations were not fully kept. And for example, when the N-terminus is shielded somehow, then it's not kept, and then the linker can also couple to it, and then you will purify, let's say, this truncation or take it with you throughout the whole process. And other main impurities are usually due to side chain reactions, which means that, for example, if you don't have a full deprotection of arginine, when you have an arginine with PBF in your sequence and it's not fully deprotected, then it will end up in your product as well. So usually it's related to side chain, deprotection or some side reactions or something like this.
I think I can add some points here. We can. So it's the sound is coming.
Sorry.
we can fine tune the SPPS synthesis to avoid some impurities like deletion peaks and aspartame formation, so that we can even further improve the PEC purity. So we successfully can remove the truncated sequences, and if we further tune the SPPS synthesis, we can even achieve the more purities here.
Yeah, sorry, that was basically the most important point, probably.
Okay, thank you. Next question from Ganesh. Do you also have the regulatory support files for this technology?
Support files means some special precautions or something that you would need, I would guess.
I guess all the documentation that you typically need for material that you use in the production of pharmaceutical material. This is what
the certificates and so on. Yeah, we do have them. Yes. Yeah.
So including all things such as BSE, TSE documents and so on and so on.
We do have this. Yeah.
Okay. Then David Fercher adding a question to his previous one. What happens then if you have a cysteine side chain, because you use cysteine in excess to block non-reacted aldehydes? Probably what happens to cysteines in your peptides? I assume you still have them protected while you add the cysteine in excess to block that.
Yes, or if I understand it correctly, if you would form a disulfide bond, then it would be reduced in the reducing step that is carried out before you release the peptide. But otherwise, yeah, for example, you can use when you use the third butyl protecting group, then it's still protected. Yeah.
Okay. Then one question, I think you already answered this. Do you know what are the side products which are still in your solution after PEC purification? This is what you already commented on also. Then there is a question from Sino Yonkes from Amsterdam. With these greener solvent mixtures, was heating used during coupling?
Generally, yeah, here we didn't use any heating, but we can also use heating as well. But in this case, bivalent case, we didn't use any heating.
Okay. Then some further questions. What is the selectivity with the lysine during the reaction with the aldehyde functionalized beads? Maybe I missed it, but how many equivalents of amino acids were used during the SPPS with this green synthesis? And what would be heating by adding pyridine in TFA in large scale?
I think I will go step by step.
Yeah, please. But so these were all questions from the same person.
I think the first question is what for the lysine, right? If the lysine protecting group is there, so it will form imine. But we, as I told you earlier, when we use cysteine, this will cleft up the lysine imine bond, and it form the stable cyclized product. So lysine won't be a problem. So it's a reversible reaction. It will form imine though, but when we use cysteine, it will be cleft off. And then the second question is, I guess, sorry.
How many equivalents of amino acids were used during the solid phase synthesis with this green synthesis?
Yes. So we used 4 equivalents of amino acids. 4 equivalents, okay. Yes.
And what would be heating by adding pyridine in TFA in large scale?
Pyridine.
Yeah.
I guess. Okay. Yeah, I forgot to mention you that what we have done is we have cooled the cocktail mixture, and we added slowly pyridine drop by drop. To avoid any excess of heat productions. Yeah, that is necessary. Okay. So you can add slowly, cool the solution, cocktail solution, add slowly drop by drop the pyridine.
Okay, then What about the instrument compatibility with the different solvents? This was a question during you explained the SpinChem RBR system. Have you noticed any disadvantages?
So we have tested the SpinChem technology with different organic solvents, for example, DMS4, and even with the direct TFA cocktail immobilization. It was quite robust and it's quite stable. And we did not observe any degradation or any rust formation or anything. It's quite stable. So you can use any solvent, any organic solvent, and it is even quite stable to like chemicals like DTT and all other chemicals as well.
Okay, then how flexible is the on-resin chemistry with PEC?
Yeah, we can use on-resin, like we can do a lot of modifications. As I told you, as I showed you, we can use disulfide formation and we can use stapling reaction as well. And we can even perform pegylation reactions. Most of the reactions are compatible with our PEC. We can do side chain modifications using our PEC technology. And then later we can purify so that we can get purified and modified peptide at a single shot.
And maybe one comment here from my side as well. Here you are even flexible, so you can either first do an HPLC pre-purification of your linker modified peptide if you really want to achieve a very high purity in the end, and then do the on-resin modification with your already very pure material. Or alternatively, you can also run an HPLC polish, of course, afterwards. And this combination will allow you to really increase your final recovery because you have a higher concentration of material with the PEC run. And then it's easier to HPLC purify it in the end if you do first the PEC and then the HPLC.
Okay. Then another question, how would you solve the problem with reattachments in the direct TFA cocktail immobilization if typical scavengers are not tolerated?
As I explained you, we can, for example, if containing cysteine and methionine peptides, this might be a problem. So we are currently exploring different cocktail mixtures, and we are currently under progress, but we can use thioanisole instead of DTT or EDT in this case to scavenge.
Okay, then is the SpinChem RBR suitable for solid phase peptide synthesis?
Yeah, those are suitable, but with our hands we didn't try, but according to the chemistry, according to the as PPS is compatible, as SpinChem is compatible for the organic solvents and chemicals, we hope that it's possible, but we have to try. We have to try. We are open for the customer projects as well.
Then some questions to the maximum size. What is the largest synthesis carried with the spin reactor and what length of a peptide? So
the length, there would be no problem as PEC can purify any length. In SpinChem also we can use length won't be a big problem. As we suggested, I shown you here, there are different reactor sizes are available. We can even go up to 10 kilogram purification of peptide in a single batch.
Oh, wow. Okay. So that one similar question, what would be the biggest possible single run scale for PepTech? So this is not especially for SpinChem, but what is the biggest possible single run scale?
Till now, I think Nadia has done, there is no limitation. We have performed 2 millimole, and Nadia showed 2 millimole scale in our simple cartridges and glasswares. Yeah, there is no limitation.
I mean, you can imagine a very big SPPS reactor, and instead of using the SPPS resin, you just load it with your modified beads, aldehyde modified beads for the PEC, and then you can really I assume you could, even with the same SPPS reactor for a certain amount of synthesis, you could process more peptide because you can reduce the concentration, you can increase the concentration, reduce the solvent that you need for the process.
So
It's basically, as Manoj said, sorry, open.
Yeah. So if you have the capacity to synthesize a lot of peptide, you also have the capacity to immobilize biotechnology.
Yes, absolutely.
Yeah. Okay. And one, obviously now the last question. Did you try to use another chemoselective reaction than oxime to immobilize the peptide for PEC?
We have tried, but oxyamine is quite robust and quite stable. So we settled it with oxyamine. We have tried with different chemistries as well.
Okay, so this turned out really to be the most, say, versatile and superior to other methods.
Yes.
Okay, so by now I do not see any further questions in the Q&A chat. So again, if you have any further questions, don't hesitate to contact us, send them by email or call us by phone. And with this, we would like to close our online workshop now. We will provide the recordings and a collection of the Q&A as a PDF. As a participant of today's workshop, you will be able to acquire a PEC peptide purification kit at reduced price. You will receive the discount code for our webshop with your next email. Thanks again for joining us. Thanks to all our speakers and also a big thank you to our quality manager Klaus Helm for taking care of the technical support. We hope you could collect some helpful ideas and information from this workshop, and we are looking forward to meeting you again in the next one in June. We will announce it on time on our homepage and via social media. Have a nice day, stay healthy, and hopefully see you soon. Goodbye.